Hi, I'm Michael Pointer, and you're listening to Abstract Bioscience, a podcast about new and exciting research in biological sciences taking place across the Norwich Research Park and beyond. On this first episode, we're going to be looking at an important issue in contemporary ecology and conservation biology, biological invasion, and something called the genetic paradox. But before we talk to researcher Ryan Brock from the University of East Anglia, a little bit of background. Since Darwin, evolutionists have known that in order for a population to adapt to the environment, it must be made up of individuals that are different from each other, like moths with slightly lighter or darker camouflage patterns, or tiger snakes with more or less toxic venom. Individual differences mean that those best suited to the conditions can be more likely to survive and reproduce. Originally, this was seen as differences in physical characteristics or phenotypes. But since the discovery that genes underlie many of these differences, we can now talk in terms of genetic variation between individuals. Generally speaking, high levels of genetic variation are associated with a population's chance of survival and adaptability, while low variation is associated with extinction risk and inbreeding. However, this presents a potential paradox when we think about biological invasions. But what is an invasion? We call a situation an invasion when a species reaches an area where it didn't already live and successfully establishes a new population. Invasions typically involve very few individuals arriving in the new habitat, and we call this drastic reduction in population size or population bottleneck. A bottleneck limits the number of genes that these few individuals can carry between them into the new habitat, and for this reason, the genetic diversity of the new population can be very low. As I already said, low diversity populations should be unfit, yet we seem to see successful invasions, where species survive and thrive in new environments, despite the population being started by a very small number of individuals. This is known as the genetic paradox of invasion. More and more, scientists are now looking at potential examples of this paradox to find out what could be going on. The author of one of these studies, Ryan Brock, has joined me today to talk about his paper on the invasion of the UK by the tree bumblebee. Welcome to the show, Ryan. Would you tell us what it is you do?
I'm Ryan Brock. I currently work as a research technician at the University of East Anglia in a project looking at kind of heatwave impacts on insect reproduction. And I've just finished my PhD. And during that, I was kind of looking at the molecular and behavioural mechanisms behind kind of social evolution in the tree bumblebee.
Perfect, thanks. And you have this great paper, which is on the tree bumblebee, which is a relatively new species here in the UK. So what do we know about how it got here?
So kind of in contrast to a lot of stories we see in the media about the kind of declining bee species, the tree bumblebee represents this really nice example of a kind of range expanding bumblebee. So it kind of first colonised the UK in 2001 in Lanford, which is a village near the New Forest. And kind of in the 20 years since, it's massively expanded in range. So it can now be found as far north as Inverness. It's crossed into Ireland. And during that time, it's kind of become one of the most abundant bumblebee species in the UK. So it's really thriving. But to date, there's kind of been no real kind of population genetic studies as to how it kind of arrived in the first place.
Great. It's nice to hear some good news for a pollinator species, because as you mentioned there, we often hear about declines in insects and in bees particularly. So do we have any clues about why the tree bumblebee might be doing so well?
So there's probably quite a few traits that contribute to its ecological success. I mean, primarily, I would say it's kind of use of urban habitats. So it's really strongly associated with urban habitats. And that's mostly because of its nesting preferences. So it makes use of kind of bird boxes and loft spaces. So it kind of really thrives wherever humans are, basically. And yeah, potentially changes in land use are one of those potential things that are leading to declines in a lot of bumblebee species. But here it could be like the other way around. If we're urbanizing areas, it might be that it favors tree bumblebees over other bumblebee species.
Wow, that's really cool. So maybe an ability to exploit habitats created by humans is helping them to establish in the UK. And so before you started working on this, what was known from a genetic perspective about this invasion?
Not a great deal. So the first kind of paper that came out about it in the UK was from Mark Brown and Catherine Jones in 2014. In there's kind of a suggestion that the UK population underwent this kind of severe bottleneck upon colonising the UK. And it's in turn, it's been picked up as like an example of this genetic paradox of invasion. And kind of more recently, another paper used a kind of molecular approach to look at kind of population density and foraging distances in the UK tree bumblebee population. And this kind of found levels of genetic diversity that were a lot higher than were originally suggested. So we knew from there's maybe more to the story than is currently known at the moment, basically.
So there was some evidence on either side, some suggesting a harsh bottleneck and some perhaps not. And the effects of a bottleneck can be particularly severe in eusocial species, can't they?
Yeah, that's definitely true. So besides all of the other negative fitness impacts that come along with bottlenecking, kind of particularly nasty in the Hymenoptera. So that's the order of insects that contains the bees, wasps, ants, and sawflies. And this is because of their mechanism of sex determination, which we know as single locus complementary sex determination. So in the Hymenoptera, sex is determined by zygosity, a single locus, and that's combined with haplodiploidy.
I remember this from way back, I think. So a locus is just a position in the genome, so a gene, and an individual can have one or two copies of the sex-determining gene because it can come from a fertilised or unfertilised egg. And unfertilised eggs get one copy, so become males, and getting two copies, which are different, means that you develop as a female. But if you're homozygous, or in other words, if you have two copies of the gene that are the same, then you develop as a diploid male, which is bad news. Is that somewhere close?
Yeah. So where you run into issues is if a population has undergone a bottleneck and all of a sudden there's really low diversity at this one sex-determining locus, what might start happening is you'll have females mating with males that share the same allele at the sex locus. And what happens there is you've got 50% of diploid offspring suddenly becoming homozygous at that sex-determining locus, and they develop as diploid males. And then you have kind of knock-on fitness effects for producing diploid males. So primarily, they're often kind of inviable or sterile, or if they can mate, they produce triploid offspring, which are inviable or sterile. And then that's exacerbated in the eusocial Hymenoptera because these are insects that live in colonies of queens and workers, and all of your workers are females. The males do nothing. The males are there to be flying sperm, effectively. So if you've got half of your worker force developing as males, all of a sudden you run into an issue with colony productivity, and there's quite well-established fitness costs to diploid male production, basically.
Okay, I see. So if a bottleneck has occurred in this expanding tree bumblebee population, then this would really seem like a paradox, given that low diversity has such negative effects in this system. But it was ambiguous on previous evidence whether the bottleneck happened or not. So when you were designing this study, what was the key question that you wanted to address?
So the idea of the paper was to kind of take two contrasting hypotheses as to how the tree bumblebee colonised the UK. So there's this first idea that it suffered this massive bottleneck. So the kind of colonisation is like a single chance event from a very small number of queens. And as a result, you'd expect kind of really low levels of genetic diversity. The alternative was that the kind of colonisation of the UK is this ongoing westward range expansion. So there's evidence to suggest that the tree bumblebee increased its range across continental Europe westwards during the 20th century. So this recent colonisation of the UK may just be part of that range expansion. So here we kind of suggest that the UK population was kind of founded by quite a large number of queens, and there's kind of constant movement from the continental mainland population into the UK population. So we wanted to kind of distinguish between those two hypotheses was the main aim of the paper.
You make it sound simple, but I bet it wasn't that easy. So how did you go about trying to tease those two possibilities apart?
First of all, so I mean, already we have a kind of panel of polymorphic microsatellite loci. So that's kind of a way of measuring kind of neutral genetic variation within a population. And that's already all developed for tree bumblebees. So here we sampled a population of workers in and around Norwich. They were genotyped, and we looked at general measures of genetic diversity, but we also used that kind of microsatellite data to run two formal bottleneck tests. So one is called a Sine Test, and that tests for a kind of relatively recent bottleneck, and the other is an M ratio test, which looks at more kind of historic bottlenecks. So maybe a bottleneck that might have happened in the last 100 generations. And there we kind of took other data from 5 other native UK species and calculated M ratios for those two. The idea here is if the M ratio for the tree bumblebee was significantly lower than those well-established bumblebee species, there's kind of that suggestion that there has been some kind of bottleneck, and that bottleneck is to do with the colonisation into the UK. Mm-hmm. So that was kind of the first part of the study. And so the second part was to kind of test levels of diploid male frequencies within the population. That's using that same set of microsatellite markers. So we can genotype the males at all these markers, we can look at them on Gene Mapper, and then if they're heterozygous at markers, that's not right, they're classed as diploid males. So, and kind of male diploidy is a fairly good indicator of bottlenecking in eusocial hymenopterans. And then from that, you can estimate the number of alleles you have at the sex-determining locus. And that was kind of the last part of the paper. We kind of produced this model where we took frequencies of diploid male production, rates of polyandry in the population, and we can plug that in, and we come out with this estimate of how many alleles there are at the sex-determining locus.
Cool, and that's presumably directly related to how many females founded the population?
Yeah, for sure, yeah. Maybe what I should have said earlier was actually how we got hold of these samples. So one of the really nice things about tree bumblebees is that their nests are super obvious. They often nest in kind of bird boxes. So one of our ways of sampling was to kind of, members of the public would have a nest in their bird box, and I would go out and collect the nest and census it and everything, and then we could take males from that nest and genotype them. One of the potential issues there, obviously, is kind of maybe survivorship bias. So if colonies are producing diploid males, and diploid males kind of impact fitness, it might be that those colonies don't get big enough to be noticed by members of the public. So to combat that, the second thing I did was to actually rear colonies from field collected queens. So bumblebees are annual insects. So they start in the spring where the queen emerges from hibernation. She founds a colony by herself. So what you can do is you can go out into the field, catch them with a big net, bring them into the lab, and rear them from there. And there you're removing that survivorship bias. And then from there we kind of sampled the males that the queens produced.
Okay, that's nice. So you've got 2 or 3 different angles that you're approaching this question from. The worker genotyping to directly measure genetic diversity, and then both the field caught and the lab reared colonies to look at the levels of diploid male production, from which you can then get an estimate of diversity. And time for the exciting bit now. Can you tell us what you discovered?
Okay, so starting with the kind of genotyping and bottleneck approach to the workers and looking at genetic diversity, we found relatively high levels of genetic diversity in the Norfolk population, higher than those that would be expected from this previous suggestion of the tree bumblebee undergoing this really severe bottleneck upon introduction to the UK. The Sine Test found no evidence to indicate that the tree bumblebee population underwent a bottleneck around the time of it colonising the UK, so around 2000, 2001 time. Interestingly, the M ratio test that we carried out found evidence for a historic bottleneck across all of the bumblebee species that we calculated it for. So that's the tree bumblebee and these 5 other kind of native UK bumblebee species. So there's this suggestion that at some point over the last maybe 100 generations, there's been a kind of population reduction. But the important thing here is that the tree bumblebee showed a M ratio that was comparable to all 5 other species. So there's no kind of suggestion there, again, that it's undergone a bottleneck upon colonising the UK. And I mean, that kind of historic bottleneck might be linked to things like agricultural intensification. It's difficult to say. But yeah, we don't have the kind of recording data, basically, to make projections about that. So they're the kind of main findings from that first kind of segment, I guess, that we were interested in.
Okay, so no evidence of a severe recent bottleneck associated with the colonisation from looking at worker bees. What about from the diploid male angle?
Yeah, so once again, kind of we found relatively low levels of diploid male production in the UK population. In those colonies collected from the field, those wild colonies, we found that a 5% frequency of diploid male production. But in the lab-reared colonies, it was higher. So we found that 15.5%, give or take, produced diploid males. So they're relatively low levels of diploid male production. You can compare them to something like the Asian hornet in France. So we know there's good genetic data there that population was founded by a single queen. And when you sample kind of wild nests there, you get a diploid male frequency of something like 50% across nests. So there's quite a disparity there. And then from those kind of levels of diploid male production, we estimate a frequency of about 21 alleles at the sex-determining locus. And that's pretty on par with native species. So kind of the overall finding would be that there hasn't been that significant a loss of genetic diversity. There might have been some loss, but not a huge amount lost.
Super interesting. So can you then make a sort of backwards prediction about how many individuals might have originally founded the population? Or do you think it wasn't a single founding event?
Okay, yeah, so we can infer from the Sine Test results using a power analysis that the founding population size for the UK, population of the tree bumblebee would have been about 30 doubly mated tree bumblebee queens or 40 singly mated tree bumblebee queens. So that's far higher than what was kind of originally stated. Of course, the alternative to that is that maybe the initial population was small and then there's just been continued immigration to supplement it with higher genetic diversity, and that's quite difficult to pick that out. And I think that's maybe somewhere where kind of future research could actually look. But I think for the purposes of this paper, it was kind of distinguishing between a massive bottleneck and a fairly genetically diverse population, I guess.
And it seems as if you managed to do that pretty well, pretty neatly, in that all of the approaches that you took sort of agree, right? Yeah,
so all of the approaches that we use come back to this same idea that the UK population isn't all that bottlenecked. So there's just a good amount of genetic diversity in the UK population, which wasn't initially the suggestion. So it's quite nice.
Absolutely, that's really nice. So what does this mean for the genetic paradox in this case?
I mean, the kind of overarching takeaway from this paper is that the colonisation of the UK by the tree bumblebee doesn't represent a genetic paradox of invasion in this case. And that's either through there being a large founding population in the first place, or potentially there being a smaller population, then that's kind of supplemented by continued immigration from the continental European tree bumblebee population. And that's kind of, as I've already said, where future research should be kind of looking.
Thanks very much to Ryan Brock, and I'll put a link to his paper in the show notes. Ryan's study on the tree bumblebee joins a growing body of evidence that what we thought were examples of the genetic paradox of invasion actually aren't. For those interested in this topic, there's a great paper from 2016 by Estu et al. Reviewing the scientific literature on the genetic paradox. I'll link to the paper in the show notes, but essentially they found that while many proposed examples of the paradox aren't real, some are. Species can overcome low genetic diversity to thrive, often through quite counterintuitive mechanisms, such as a large reduction in population size actually increasing fitness by removing bad genes from the population. The authors of that review also say that the only way forward is to do, quote, more science. With more research, we can uncover more about the conditions under which species become invasive and use what we learn to prevent invasions that threaten native biodiversity. That about wraps up this episode. Thanks to Ryan Brock for sharing his work and to his co-authors for writing it with him. I'll be back with more science for you on episode 2 of Abstract Bioscience very soon. In the meantime, show notes, details about the podcast, social media accounts, and links to information about the Norwich Research Park can be found on the Abstract Bioscience website at abstractbioscience.co.uk. The show is written and presented by me, Michael Pointer, and you can contact me at abstract.bioscience.podcast@gmail.com. We're on Twitter at abstractbiosci, Instagram at abstract.bioscience, and our excellent theme music is by Phoebe Troop. Thanks for listening. Catch you next time.